It's a lot of fun to be here and to present. I will keep this extremely informal. Interrupt, ask questions. If I'm too fast, stop me. And I will give you my personal view of, as Dana says, an end user of a technology that I have seen develop over the last, I don't know how many years, starting with the first experiments that I did as a postdoctoral researcher while I was working at the Whitehead Institute with Rudolf Jenisch, where we were interested in human pluripotent stem cells, which are really cool cells. They are pluripotent, which refers to their ability to differentiate into, as far as we can tell, any cell type of the human body. And based on that, they have great interest, therapeutic interest, research interest. And we were tinkering around with these because at the time where I started my postdoctoral work, Yamanaka just had published the process of reprogramming, where you could take a skin cell and convert it by expression of reprogramming factors into this pluripotent state. And all of a sudden, we had a genetic system. We had a system where you could take a fibroblast of a patient that had a genetic predisposition and generate a pluripotent cell that then had the ability to, while having this genetic predisposition, to generate any cell type of the human body. And that, for the first time, gave material that is really interesting to study. For example, it allows you to derive a neuron of a patient that has a neurodegenerative disease, and you can investigate what is the genetic predisposition that is in the affected tissue present and leading to the disease phenotype. That is, at least, was the idea of why iPS cell technology had such a great impact. So, and you can see how that is for neurodegenerative disease particularly interesting because, for example, in a Parkinson patient, the last thing that you would do in order to do research is biopsy from a Parkinson patients where the dopaminergic neurons are lost during the progression. So research was really stuck and limited to doing Parkinson research in HeLa cells, where this is not that long ago. This is 10 years ago where this was common practice. And luckily, with these new tools of reprogramming pluripotent stem cells and now genome editing, we don't have to do that kind of research anymore. Okay, so the setting is we have a cell type that can that gives us research tools and cellular experimental system that we are interested to investigate. And while I was in Rudolf's lab, we teamed up with Sangamo Bioscience to develop genome editing technologies in this cell system, because it became very clear that working with these cells have their limitations. And that is mostly they, at the time, were not a genetically accessible tool, meaning that we could not genetically manipulate them. And so over the turn of a few years, we used zinc finger nucleases and later on a technology TALENs to show that these cells can be genetically engineered to generate reporter cells, to delete genes, to overexpress genes, and to perform gene corrections. And I will show you a few of those examples because they are really what we thought were proof of concepts for this technology to actually turning this from a non-genetic system into something that allows us to ask very pointed and very clear research questions. Before I'm going into that, I will quickly give you a summary of where these human pluripotent stem cells come from and a quick background into developmental biology. And most of you are familiar with this, of course, where human pluripotent stem cells are isolated. This is a timeline after fertilization of a mouse embryo, and I'm using the mouse embryo for an example of how pluripotent stem cells are isolated at the time where the first cleavage stages of the embryo are completed and the morula is compacting and then forms into the first differentiation step where we have an blastocyst formed where an extra embryonic tissue is formed that will eventually lead to the implantation of the embryo into the mother. And then there is a specific cell type that we call the inner cell mass. And this inner cell mass, in the mouse development, this happens at around 3.5, will develop what we call an epiblast. Since these cells have undergone their first differentiation step, and they already developed into two tissues, these cells are considered pluripotent, and this is in contrast to cells that are at the earlier step of the embryo development, where we call them totipotent. Totipotent means if you would cut this embryo into half and you would allow these two cells then to develop, they have the potential to form a full organism. A pluripotent stem cell is already restricted. It can only form the tissue of the embryo, some additional tissues, but it can't make the extraembryonic tissue anymore, meaning that it will never form by itself a full organism. And I think that is very important for the ethical debate of what does editing mean in human pluripotent stem cells. It illustrates that the ethical concerns of editing in human pluripotent stem cells are comparatively little to editing events that would be preceding fertilization or during the totipotent state. That makes, just to provide some context, that what we are doing is at a developmental state that cannot make a human being by itself. Okay, so the introduction or the first lecture by Jacob was actually in a perfect setting because that means that I don't have to review much of the events that happened after a double-stranded break is made. The thing that I will point out here is that what I will review today and what we are currently using in my lab are almost exclusively homology-mediated repair events. And you can think about this as, and you know that after a double-stranded break is made, in this case still a cartoon that uses a zinc finger. By now my lab has completely moved to using Cas9. After a double-stranded break is made, there are two pathways that can repair this. One is the non-homologous end joining that is error-prone and can make insertions and deletions, and Jacob described that today. And the second one is where you can use a repair template to, that has homology to the sites of the double-stranded break to repair this. And this is a homology-mediated event. And we think of the second repair event as driven by synthesis-dependent strand annealing, where one side of the break is resected. Strand invades in the homology donor, is extended by a polymerase to the point where you synthesize new sequence that is homology to the other side of the double-stranded break. This can be dissolved by finding this homology on the other strand. You can prune overhangs that are potentially there, and you can fill in gaps and thereby repair your double-stranded break by homology-mediated event. NHEJ editing is easy, okay? NHEJ as editing is highly efficient, and that is in striking contrast to editing by designed specific modifications that are homology-driven, at least in human pluripotent stem cells. I will exclusively review the experiments that we did that are utilizing, as far as we can tell, homology-mediated strand annealing. And the three proof-of-concept experiments that I would like to go through is the establishment of a safe harbor locus that allows you to overexpress genes in human pluripotent stem cells. We will quickly touch on the generation of a cell type specific lineage marker, and the experiment that I will start out with is how to make a point mutation in human pluripotent stem cells using homology-mediated repair. The last experiment is the coolest experiment of these three. It was done by a very good friend of mine, Frank Saltner, who was a co-postdoc with me in Rudolf's lab. And Frank is interested in Parkinson research. And the iPS field ran into a really big problem. So I explained to you how it is really interesting to investigate the phenotypes of an iPS cell and then a Parkinson patient-derived neuron to investigate what maybe the disease underlying feature of the cell is. And Frank did this and set up the experiment, made several Parkinson lines. However, one thing that became very early on very clear is that the genetic variability between individuals can supersede the phenotype that you are actually looking for. So if we consider that we are different from each other by approximately every 1,000 base, you can see that the genetic diversity is really challenging this experimental approach, where we are assuming that we can infer a genetic defect in a patient sample and can predict the cellular behavior in a dish. And Frank came up with the perfect solution for this experiment. He said, okay, if we can take a patient fibroblast of any genetic background we can take this fibroblast and we can reprogram it into an iPS cell. And then he hypothesized, okay, this problem of genetic diversity within the human population he's just going to solve by repairing this mutation. And by repairing this mutation, he establishes now a cell line that exclusively differs only at the mutation point of interest. And then you can take these cells and differentiate them into cells that are isogenic, perfect matched cells, and you can infer the phenotype just by comparing these two cell lines. And you can do the same experiment following the same rationale and logic, where you take a wild-type cell that could be an embryonic cell or a cell out from a non-affected patient, and you can introduce the mutation, and then you again have an isogenic cell line, and you can differentiate these and investigate the phenotype. Frank demonstrated this on a gene, alpha-synuclein, which in a heterozygous setting, specific mutations in alpha-synuclein predispose to familiar forms of Parkinson's disease. And he introduced a specific mutation using zinc finger nucleases, cutting at a specific base. And dependent on the homology repair donor, he either inserted this mutation in human ES cells or corrected this mutation in patient-derived iPS cells, generating isogenic cells. And this is just a sequencing read of a heterozygous cell line that he generated in a human ES cell line carrying this specific mutation. And he went on to differentiate these cells into dopaminergic neurons, which are the cells that are affected in Parkinson's disease. And I think this is really cool, because it shows that at least in a cell line, we can use genome editing to refine the human genome with base pair precision and overcome one of the key challenges that we were facing in stem cell differentiation problems, which is inherent to the genetic diversity. So this is a workshop, and I thought, since I like this experiment so much, I'm going to give you 3 simple rules of how to make this happen if you wanted to do this. And they have to do with the repair mechanism that underlies this repair. And it's very simple. If you want to have a successful editing, you stay as close as possible to the double-stranded break as you can. Okay? So in human pluripotent stem cells, editing is significantly less efficient than in cancer cells. And we don't have the luxury of picking many guides. If you go too far away from the double-stranded break, and we think that this has to do with the amount of resection that happens after the double-stranded break is generated, your editing will fail. So if you want to introduce a point mutations, you stay as close as possible. You make your break as close as possible to the change that you want to make. The other thing that is very intuitive based on this repair reaction is if you want to employ this particular repair reaction, you see that there is one synthesis step, and this is directional. So if you want your repair to go through this repair mechanism, then it is, and you want to edit with a single cut, it is very helpful to introduce mutations only to one side of the break. If we look empirically, this is heavily favored over having mutations on both sides of the break. The other thing is also becomes the other third rule that I think is a little bit more complicated, but try to think through with me here. We are synthesizing, starting at this point, new DNA that is based on the sequence of our donor plasmid. And we are going to continue doing this until a point where we synthesize DNA that is homologous to the other side of the double-stranded break. If we had additional homologous regions within this gray thing, which could be our GFP that we want to integrate into the genome, then we would dissolve this prematurely. We would find homology to this side of the break before we actually wanted to do it, because we only went halfway through our GFP. These stretches are, again, the homology there has to be quite exact. So if you don't have to start predicting always their homology. But if you have repeats, this is actually becoming a concern. And it also means that you cannot, if you think this through, make multiple mutations that are far away from each other within a specific donor. If you have questions about this point number 3, many editing events fail because of this, and I'm more than happy to answer emails that are speaking to this particular point. Okay, this is a table, and this table is to show you 2 things. These are editing efficiencies introducing specific point mutations in different targets, in human ES cells, within my lab. And what we learn there is that this is still extremely low using this technology. And the reason why this is so low is because human pluripotent stem cells are not cancer cells. They still have a fully functional DNA damage checkpoint. Many cells that have a double-stranded break or have double-stranded break plus potential off-targets will just die. Repair mechanisms are probably relatively efficient by repairing stuff in a way that we don't want. And so if we look at the number, these are just 9 randomly picked experiments that we did in the lab. We get picking 50 individual clones, human ES cell clones, growing them up, and then genotyping. We get one in this experiment. But there are many experiments where it actually fails. Okay, so there is much room of improvement. If we were to, and I probably should have done that, here would have added how often is this locus actually genetically modified in a way that we didn't want by NHEJ, insertions, deletions. It would be for almost all of these in the 10% range. Okay, so the ratio between HDR, which is this one, and NHEJ is heavily skewed towards NHEJ, and that is under research. A lot of research is going into how to make this much better. Okay, so yeah, hit me. Usually it's a point change plus a restriction site to detect a silent mutation in order to make it easy to genotype, because otherwise you have to sequence, and that's just expensive. Yeah.
Plasmids or oligos?
So this is most of this is plasmid. Most of this is plasmid. But so if the alpha-synuclein experiment that I showed earlier was done actually plasmid and oligo, and the efficiency is not much different. So in Parkinson-derived iPS cells, it was 2 in 200, which is this efficiency, oligo. So there are 2 things to take into account. iPS and ES cell lines will accumulate a history over time in culture, and they will become a little bit more cancery and easier to work with over time. That is something that you want to avoid, because at some point they are cancer cells or teratocarcinoma cells. So if it ramps, if you see a history and all of a sudden your editing becomes better each week, I would throw those out because it's not good evolution in addition. Okay, I want to speak I want to bring up this slide. And I have this slide for 2 reasons. I want to discuss with you next why it is important to genotype properly and to genotype correctly and not just by PCR. That's one thing that I wanted to bring up. And then I wanted to bring up how do we deal in the scientific community with the vast amount of alleles that will be generated by NHEJ. Okay, so, and I'm going to play devil's advocate here. Mouse genetics used to be very clean. We had mouse genetics, we had mouse strains, and they were inbred. You would delete an exon in a mouse, you would share this mouse with another researcher, and the experiment could be reproduced in 2 different places of the planet. Okay, so now in the new world of Cas9, it will most likely look very different. We will have to understand every aspect of, is an insertion into a gene and a deletion the same thing if we generate hypomorphic alleles, right? So if you have a loss of function, if a message is generated to nonsense mediated decay, is that the same if the exon has a 15 base pair deletion or not? What I'm trying to point out here is that if you are operating with NHEJ, you're in a bind to demonstrate that this is a genetically sound allele. Most events in using just NHEJ will generate compound heterozygous events where, yeah, if they are out of frame mutations, fine, but you have to make sure there's no splice variant, no, okay. One argument, one way to go back to good old mouse genetics is Cas9 based and is shown by this experiment. And this is where we have an exon of a gene, and it's a southern blot for an exon where we generate a knockout by just cutting with 2 Cas9s and remove an entire exon. Okay, so this is on both alleles generating the same genetic modification, and it makes it relatively easy to then predict, okay, this is an essential exon, and we don't have to deal with what happens if there is a premature stop in a gene. Do I all of a sudden select for other events that could be confounding? I'm just saying that the diversity generated by NHEJ-mediated editing will be very confusing to us, and we will have to sort through many arguments. Oh, my Cas9 edit is different from your Cas9. So genotype correctly. And sorry, here going back to targeting efficiencies, I want to make the point again that it's cell type specific. It's very low in human ES cells. NHEJ is much greater than HR. And there are large differences between cancer cells and primary cells. Primary cells, there is the nice exception for primary cells seem to be hematopoietic stem cells. CD34 positive stem cells seem to be editable very well. Okay, so I'm going to now go through the other two experiments that I wanted to describe to you. One is the establishment of a safe harbor locus by locus for controlled gene expression. This became relatively clear that we needed in human pluripotent stem cells a system to deliver genes. And now we are not talking about the modification of one specific allele or one specific gene, but we wanted to find a way to overexpress genes in human pluripotent stem cells. And in mouse genetics, we had these tools at hand. Most of you are probably familiar with the ROSA26 locus, which is a safe harbor locus that is an easy targetable locus to overexpress genes in an ES cell or then eventually in a mouse. ROSA26 or the collagen locus are good loci. When we started out, that was not available. And we decided to use what is called the AAVS1 locus in human ES cells to develop such an analogous system in human ES cells. The AAVS1 locus is the preferred integration site of adeno-associated viruses and has been predicted by others to be a locus that can be targeted without variegation and without silencing effects, and without adverse effects in a homozygous setting. And so to test that this is actually the case, that we can target this, we designed a donor that had homology to a zinc finger nuclease cut site that we introduced after exon 1 of the AAVS1 locus. And we either integrated a splice acceptor puro site at this position. This is a gene trap that picks up the message of exon 1, splices it to puro, thereby renders cells puromycin resistant. Or by integrating a PGK puro, which is an autonomous selection cassette that drives expression of the puro using this PGK promoter. Both of these targeting events were highly efficient. 50% of all clones were correctly targeted by plasmid delivery of the donor plasmid and just by expression of zinc finger nucleases. These efficiency are exactly the same if you use a TALEN or if you use a Cas9 plus minus a few percent. And we have directly compared that.
Sorry, that's among the puro-resistant clones.
That is among the puro-resistant clones. If you look at homology-mediated events that are not selected, then it is below 1%. It's below 1%. So that's a very good point. We are here selecting for homology-mediated event by using a trick by integrating a selection cassette. If we were not to do that, this event would be in the 1% range. Yes?
If you omit the nuclease, what kind
it never happens. It never happens. So There are a few reports that you can use just homologous recombination in human pluripotent stem cells, and the efficiencies of those are 1 in a million. Okay, so this me saying it never happens is exaggerated. It might happen. We never observed it. We did those experiments, and we never see it. Generally, so these experiments, the best experiment ever done in this regard was the first one also by Zwacker and Thompson, who targeted the OCT4 locus, a locus that we also targeted. And their efficiencies were extremely low. I think they, in total, generated only 2 clones.
The next question is, have you ever looked at where these off-target insertions are going? And is it related to an off-target effect of the nuclease?
So you're saying, where do the others go?
Right.
Yeah, that's a very cool question, and I will show you that. So in many cases, the aberrant targeting events go into the same locus. And they are mediated, and I have a slide about that. They are mediated by an event where we do homology-mediated repair. And then we synthesize the DNA, we synthesize the selection cassette, and then at some point we decide, oh, let's fix this with NHEJ. Let's not search properly for where the other side of the double-stranded break is, and you just patch that back into the break. And the easiest way to prove that that is the case is you hybridize with an ampicillin. If it's done off a plasmid, all of a sudden you see ampicillin in the AAVS1 locus. Okay? And we sort those through. We sort those out using Southern blotting to make sure that these events are not accounted for as correctly targeted. Right? Pure resistant here and correctly targeted is not the same, because those aberrant targets that are doing HR and NHE, they are aberrantly targeted. So we eliminate those. And I have a slide, but this is a very good question. Because for a gene trap, of course, you're expecting much higher targeting efficiency, right? Yep.
And another question, in those cells, what is your ratio of biallelic versus monoallelic integration?
Yeah, that's a good one. I have a nice overview at some point. In a gene like OCT4, which I think we have, do we have this here? This is an essential gene. We never get a homozygous clone. If we target the AAVS1, this is now with talents, but trust me, zinc fingers is exactly the same. Homozygous clones is 21 homozygous, 34 heterozygous, overall targeting efficiency 57. So this is number of clones. We picked 96 clones for the PPR. This is the AAVS1 locus with a splice acceptor puro, and we get targeting efficiencies of 57%. If we use a PGK puro, we get 52 heterozygous, 13 homozygous. So you can directly homozygous a locus with one step. Okay, more questions? Yeah?
Yeah, this is kind of related to the earlier question, but the relatively similar rate of correct editing in your puro-resistant clones, whether or not there's a splice acceptor, which is autonomous, that would suggest that the plasmid does not frequently integrate via non-homologous. Do you agree with that?
So the likeliness of random integrants of the plasmid is very low. It's very low, and most of the aberrant targeting is driven by the event that I just described, which is the, yeah. Okay. This is a slide to demonstrate that iPS and ES cells are behaving exactly the same. Here we have the heterozygous, the homozygous number. This is the efficiency for iPS cells, and they're interchangeably ES cells versus iPS cells. So this targeting experiment only puts a puro in there to select. That's relatively boring, right? What does it help us to put a puro into a locus? This is the key experiment. This is the key experiment where we can show that if we integrate a very strong promoter, a CAX promoter that drives GFP into this locus, either heterozygous or homozygous, we get uniform expression in these cells. If we differentiate these human pluripotent stem cells in this case ES cells, into what is called a teratoma. This is the subcutaneous injection of targeted ES cells in immune-compromised mice. They will spontaneously form a tumor called a teratoma and then differentiate in this mass. And then you can identify different tissues that are coming from these ES cells, and we can track back, are these tissues still GFP positive? And we find that cartilage tissue, intestinal tissue, smooth muscle, neuronal tissue all retain GFP expression, arguing that this is a locus that can be used to overexpress not only genes in the ES cell state robustly, but also by differentiation. Later, so this is dependent on a relatively strong promoter, but if you use this promoter, you will get robust expression in downstream tissues. This can be controlled temporarily by modifying the GFP expression, putting it under a promoter that is doxycycline inducible. If you do that in the ES cell, you get robust expression. And this robust expression is completely dependent on doxycycline. If you don't add doxycycline, then the cells are GFP negative. However, increasing amount of doxycycline, which are represented by this curve, gives a dose-dependent increase of GFP expression in the ES cell state.
Have you tried the doxycycline-induced expression in differentiated cells? How does that
No, have not.
We have seen some cases where it seems to get silenced.
Yeah.
In certain
Yeah. So that's
CAX seems to be more
CAX is much stronger. That's why I pointed out that the CAX promoter is beneficial. We use this system a little bit further to show that you can actually generate compound heterozygous within a single targeting step. You could call this the first multiplex experiment, where we use two different donors to target one allele of the AVS1 with a TET-inducible H2B GFP, and then this TET promoter is dependent on the expression of this M2-RTTA, and we delivered this M2-RTTA by targeting the second allele of the AVS1 locus, and H2B GFP then will lead to very nice. Let's see if I can find this. Here it is. Staining of human ES cells undergoing, let's find one, undergoing mitosis. Oh, give me one more. It's not bright enough, I'm sorry. There are many, but maybe they're all in the background. You can follow cells going, human ES cells going through mitosis. Yeah, on my screen I see many more cells than these three there. I'm sorry about that. The key experiment here, and what I'm trying to show you, is that these editing events are independent, and that you can target chromosomes at the same time using co-delivery of two repair plasmids at the same time. Okay, so now I would like to quickly talk about the generation of a cell type specific reporter gene. At the time where we made these Parkinson iPS cells, we thought, okay, one rate limiting step to actually analyze these cells is efficient differentiation into dopaminergic neurons, which are the cells that are affected in Parkinson's disease. And so the idea was to use genome editing to facilitate this process and to genetically mark a transcription factor that turns on during the differentiation into dopaminergic neurons, which is this gene called PITX3. And the idea of this experiment is relatively simple, where you integrate a GFP into the ATG of this PITX3 gene. We had a selection cassette, a PGK promoter driving a pure myosin, and then a polyadenylation signal that is flanked by LOXP site that after the targeting event can be excised, and the reporter would be a reporter that only reports on the transcriptional status of the promoter element, and then the GFP would polyadenylate after being integrated into the ATG of exon 1. And this targeting worked, and it worked actually quite nicely. The reason why I'm bringing this targeting up is because of the significant amount of aberrant targeting events that are specific to the PITX3 locus. And this is shown here. So if we the way that this experiment is done, we made this donor. This is again a targeting that is done with talons, where this cut is made, and then we provide this repair template, and we grow up human ES cell colonies and genotype those colonies for correct integration into the PITX3 locus. And we genotype these with two different probes. One is an external probe, and one is an internal probe. And the external probe will identify heterozygously targeted clones, which are shown here. And this is a 5' probe on this side. However, if we use an internal probe, so a probe that will hybridize to the donor plasmid that bears homology to repair this, we find that there are many additional integrations, despite the fact that on this side they are correctly targeted. If you genotype these cells by PCR, they look completely fine. And they look completely fine because the additional integrations that are happening in you see these additional bands that are aberrant, are the product of what I described, where there is a heterologous targeting event using homology on one side of the break, but aberrantly integrating plasmid DNA on the other side. And that is something that we see for most of our targetings. If you carefully Southern blot for trying to identify how does this locus actually look after editing, then these are the frequencies with which we see these additional integrations. And for the AAVS1 locus, for example, we see up to 20% of the events, sometimes even higher, are corrupted by this heterologous integration. And the same is true for the PITX3 locus. This would be extremely difficult to detect by PCR, because depending on where your breakpoint is, your primer still might anneal to an inside and an outside primer and give you a size that you would judge by size, oh, this could look all right. The only way to tell is for additional sequence integration by either long-range sequencing or better by Southern blotting. Okay, are there questions to this? Okay. All right, so this is the technical part. How much time do I have?
So 25 minutes.
25 minutes. Perfect.
Can you do that?
Absolutely.
Dirk, actually, I had one question
Yeah.
for you. Obviously, it would be nice to be able to have more safe harbor loci like AAVS1.
Yeah.
Have you tried other ones?
So there, Gordon Keller, I hope I got that right. Gordon Keller reported on the identification of the ROSA 26 locus and the synthetic region that corresponds to the ROSA 26. I personally have not targeted it, but his data looks very good. I would suggest to going for that. And then there is the CTL. Does somebody know? There's a third one that was proposed by somebody at Sloan Kettering where the data also looks good. And I can fish those out and find them.
People use CCR5 also.
Okay, yeah. So there are some out there. And if you're interested, I find the citations for you. There is a good review by somebody at Sloan Kettering who wrote what are the requirements for a safe harbor locus. I think the AAVS1 locus is a good expression locus. I personally am not a clinical person, and I don't. Sangamo is targeting currently, I think, into the globin locus for a safe harbor locus. I've heard some data about that. So there might be a difference between what you would like to accomplish by a research experiment. I want robust expression in an ES cell and differentiated cell versus a safe locus that could be clinically relevant. Okay, so this was all very technical. Are there more questions? Okay, I'm going to switch gear and I'm going to tell you about what we use all of this stuff for, because this is all in the past, and you saw that this is done with technologies that are ancient, like zinc fingers and talons. So let me give you a brief overview of what my lab is doing. When I started my lab, I was in this weird position of coming out of technique development and how do you engineer a human stem cell genome and how does that work. But that became extremely fast, extremely competitive, right? Cas9 basically threw everything over, and I was very happy that I decided, okay, I will not be part of this technique development craze, and I decided I'd go back to some biology that I care about, that I like, and that I learned about during my PhD training. And that is telomere biology. And of course, this was not completely out of nowhere. There is a question that I'm very fascinated with, that I find very interesting, that can, in my opinion, only be addressed in human pluripotent stem cells, and that has to do with telomeres. Okay. So telomeres are the end of linear eukaryotic chromosomes. They have two functions. They protect the genome and the natural chromosome end from being recognized as a double-stranded break. This is now where we talked so much about double-stranded break repair. An obvious thing that if you are a double-stranded break, you don't want to artificially repair that if you're a natural chromosome end. And then the second thing that the telomere does, it provides a buffer sequence for sequence loss, for example, through nucleolytic attack or through the end replication problem. The sequence at the end of the chromosome, this telomere, is made out of tandem repeats of the 6-mer TTA GGG. And this sequence is bound by a protein complex called the sheltering complex that interacts with one of the coolest enzymes around, which is telomerase. Telomerase is an enzyme that can make these TTA GGG repeats. It's a reverse transcriptase that can extend this 3' overhang using an RNA template sequence that's called TR and a protein called TERT. And it's really cool because this enzyme telomerase, its expression is restricted to stem cells. It's only on in stem cells, but in most differentiated cells, it's off. And that's already a good argument to study this in a stem cell system. The second really interesting thing about this enzyme is that it's or differentially regulated in mice. Mice, almost all cells are telomerase positive. Telomeres are extremely long, and they retain this enzymatic activity even in fibroblasts, for example, which are somatic cells. So the mice, to study the processes of telomere biology, are actually quite some outliers, and we don't touch those for that reason, because they have very long telomeres. The reason why it's interesting to understand how this enzyme works is because it is the regulation of telomerase has profound outcomes for a human cell. In a human pluripotent stem cells, telomerase is on, telomeres, this repeat at the end of the chromosome, are stable over time, and they don't shorten. Once telomerase is transcriptionally silenced in somatic cells, telomeres gradually shorten and at some point fail to perform the two key functions that I just told you. They don't protect the chromosome end anymore from being recognized as a double-stranded break, which is illustrated here. This is a staining for telomeres in red. And then here for a DNA damage protein called H2AX. And when telomeres become very short, all of a sudden they become deprotected, and they localize to the sites of the telomere, double-stranded DNA damage proteins. And this leads to a cellular response called senescence, and cells will arrest and stop dividing. Okay, so proliferation stops, and this barrier is called the Hayflick limit and is considered a tumor suppressor mechanism. That's also intuitive, because it means that a cell in our body that is differentiated has a proliferative capacity, and it will not supersede this proliferative capacity. And that is hardwired by telomeres. This proliferative stop can be overcome by checkpoint inactivation. For for example, loss of p53, and then telomeres will actually shorten a little bit further. This will lead to chromosome fusions. At this point, the chromosome end is repaired, artificially fused to each other, and this leads to dicentric chromosomes. If you try to put those through mitosis, cells will eventually die. And this step is called crisis, which is thought to further exacerbate the cancer phenotype of cells that are entering this, because all of a sudden you get genomic instability by breakage bridge fusion cycles. Okay, now telomerase gets into the game. We were telomerase negative. This started all of this telomere shortening. In 90% of all tumors, or more than 90% of all tumors, this telomere shortening is stopped by the reactivation of telomerase. Telomerase turns for some reason back on and allows tumor cells that are arising at this point of crisis to stabilize their telomeres and live indefinitely. And this is shown here by, this is a telomere length plot where we're looking at the terminal restriction fragment of chromosomes. So we take the genome, we cut it into small pieces with restriction enzymes that normally do not cut in this, or that do not cut in this sequence TTAGGG. And then we just run it out on a southern blot or on the blot, and we hybridize with a probe for TTAGGG. And here you see that in a human tumor cell line, telomeres stay stable over time. Okay, so this is the, sorry for the long introduction, but I'm going to tell you one story why we have all of my lab is evolving around this question, and I will tell you one story about this. But before I do that, I want to point out how critical it is to actually understand this. So in a stem cell, we start out with the telomere reserve that predicts how often a cell can divide, and it functions to set up a tumor suppressor mechanism. So if we envision that telomeres were aberrantly long in a human being, then this tumor suppressor checkpoint would kick in too late. And cells would get a proliferative advantage and they could divide much longer. This is actually seen. There are mutations in telomerase, in the telomerase promoter, that will lead to increased expression, germline mutations, that lead to longer telomeres, and these patients are developing cancer with an onset of 40 years or so, invariantly, if you have this familiar form. I find that really fascinating. On the other hand, if you were to start out an organism with telomeres that are just a little bit too short, then you don't have the proliferative capacity to make a full functional organism. And that is a disease that is called dyskeratosis congenita, where mutations in telomerase that dysregulate just telomerase by 50% all of a sudden leads to bone marrow failure, again invariantly with the onset of 40 years. So that's the challenge that we have to study here. Very tight window of regulation and within a very long period of time. And we need to find genetic models that are that allow that. I'm going to skip through this. Okay, so last year I had the privilege to speak at this workshop last year. I decided to this year to talk about a different experiment that I very much like and that highlights the power of genome editing. You have to bear with me, unfortunately, through a little bit of molecular stuff in order to. So I told you about a protein complex that is called this shelterin complex. And this shelterin complex sits on the telomeric DNA, and it communicates with this protein called TERT, telomerase, that normally elongates this 3' end. So far, so good. So this is a very simple cartoon. When I started out my lab, we wanted to know about the genetic requirements for this particular interaction, for the interaction with this protein called TPP1, which is part of this shelterin complex with telomerase. And we wanted to know how important is this interaction for telomere maintenance in human pluripotent stem cells. And so we performed a very easy experiment where we deleted the interaction domain within TPP1 using genome editing, and where we made a homozygous deletion mutant using homology-mediated repair to delete just 6 amino acids within TPP1 homozygously, and to ask what happens to the interaction with telomerase. And we found that this mutant behaves exactly like a telomerase knockout cell. This interaction is 100% required for telomere maintenance, and if cells don't have this interaction, cells will die. And that set up the experiment to ask, okay, what within this interaction is important to mediate TPP1 telomerase interaction. The protein the interaction domain that we deleted is this 6 amino acid repeat here. So we had cells that are, I'm going to do this. We had cells that were deleted for this TPP1 delta loop, which is the interaction domain with telomerase. And we next ask, what happens if we complement this deletion cell line with different alleles of TPP1? And in order to do that, we used AAVS1 overexpression, and we complemented this mutant phenotype with different forms of TPP1. And I think this is a really cool result, where if we are delete for TPP1, telomeres are short. And if we complement this phenotype with the wild-type form of TPP1, telomeres elongate gradually back to what they're supposed to be in a wild-type cell. So we are going from a null phenotype that is deficient for interaction with telomerase, and we complement this phenotype by putting back the wild-type form of the protein, resetting telomeres back to a wild-type length. And then we went through a panel of TPP1s and asked, can every TPP1 that now has additional mutants that are not, this is wild-type, these are mutants within TPP, can they perform this assay the same way? Can they rescue a null phenotype to the same extent as the wild-type version of this protein? And as you can see in this, there are different behaviors of different mutants, and some of them actually do reset telomeres eventually to wild-type. However, one mutant did not. One mutant fell out of this screen where it did not faithfully complement the interaction, the deleted. And this is a mutant that in the structure is far away from any of the interaction domain, leading to the hypothesis that it actually does something different than just mediating interaction. The reason why I'm telling you this is to highlight an editing experiment, where this is a very convoluted complementation experiment. We first make a mutant, we complement it, and we ask, is it fully intragenically complemented or not? It generated a hypothesis to look at this specific amino acid. And then we decided, okay, let's make this amino acid change now homozygously in a wild-type background, not in a delta loop background, but in a wild-type background. And this is the experiment shown here. Wild-type telomeres are maintained stably over time. This is human ES cell. Telomeres are at around 9 kb. However, if we made this mutant that we hypothesized based on this previous screen to carry this homozygous mutant, telomeres are short, but they are short, stable over time. And this experiment was done by genome editing, specifically at this position, at this 104 position. And now we were in a problem. We had got only one of these mutants. It was one of these in the table that I showed you earlier was 200 clones picked. Only one of them was homozygously targeted or correctly targeted. So we had a problem here. We had one mutant that behaved exactly as we predicted. But how do we prove that this is not an outlier? How do we make sure that this phenotype is actually what it's supposed to be? And that's the cool thing with Cas9. We took this mutant. Luckily, the mutant itself retained a PAM and retained a guide RNA binding recognition site. And we could just revert the mutant. And we could take the mutant that was found in this bizarre screen from amino acid 104, from L to A, short telomeres, and we just went back to the wild-type sequence, and telomeres reset. Okay? So the reason why I'm going through this experiment is to highlight that we can do clean genetics, and we can test we can test hypotheses, and we can actually proof almost that this has to be, this is not random behavior, right? So if you're ever worried, you subclone the cell and you want to know, is my edit really what I thought it is? Make the revertant. Make the mutant, edit it back to wild type and show that you restore functionality. Complement your phenotype, right? This experiment shows that if you have a knockout that has short telomeres, this is wild type, this is the mutant, this delta loop, telomeres are short. If you complement it with the wild type cDNA, you will reset telomere length. And if you weren't, maybe something is wrong, right? If you then derive a hypothesis of, you don't have to remember any of these numbers, but you see that all of these 104s are short here, right? So they didn't complement the phenotype properly. And we took this as a discovery tool to then hypothesize, okay, what happens if we make this mutant? We edit it. This is all done by Cas9. We got a heterozygous. The heterozygous is actually behaving as wild type. We got one homozygous event, sequence that. Telomeres are short, but you get only one. And an N of 1 is really hard to publish. But if you show that your revertant is wild type, then I think you're fine. That's what I got. Yeah. Sorry, there was like kind of a lot of stuff in here. Yes.
When you're introducing point mutations in the donor, are you typically mutating the PAM as well with a synonymous mutation? Or are you relying on mismatch and intolerance?
Yeah. So most of these edits are introducing additional mutations just for the reason of making a restriction site to be able to detect easily the modified allele. If we can destroy the PAM, we do, but we don't make it 100% of a requirement. The biggest problem currently is not the biggest problem currently is compound heterozygous. One being correctly targeted by homology-mediated repair, the other allele destroyed. That is a cell line that is really hard to work with because you don't know what this allele is, right? And that's not what we want. So we throw all of those away. Usually don't work with them because it's really difficult to work with this cell line and have it. So does that make sense? So I don't think the PAM ablation, if you can do it, do it. The other rule, if it doesn't violate the rule that you only want to edit to one side of the cut, then go for the PAM ablation, yes. But it's not absolutely necessary.
So are you southern body 100 different cell
Yeah.
lines every time?
Yeah. But here's the point. That is the beauty of human pluripotent stem cells. And I'm going to make a shout out here for cell biologists. This is really an entity that lives indefinite in culture and can make a lot of other cells, right? So other different cell types. So subcloning with these guys is super easy. This is not like fibroblast subcloning where you need conditioned media and then they grow or they don't grow or limited dilution. You throw them on the plate. We do everything on feeders, which keeps the IPS or ES cells super happy. You just pick them. Done. So 200, if you're well trained, is 2 hours, 3 hours picking, and then they grow, right? You just change media. 10 days later, you make a replica plate, and that replica plate is already size enough to southern, and that's one gel, right? That's 27, 81 per gel. So if you have a workflow for it, trivial. It looks complicated, but it's really easy.
I have a related question, which
Yeah.
is, because you can clone so easily, if you ever do whole genome sequencing, the complementation is a good way to assure yourself that the phenotype you're seeing.
Yeah. So I'm gonna answer this very indiplomatic.
How unusual.
Yeah, so genome sequencing was available prior genome editing. And one could say that the whole genome sequencing should have been done much, much earlier. People have done the experiment that you're asking for. It's currently, the ends are still very low to actually make conclusions of how much off targets are there. I think it should be done. I personally don't have the money. If somebody gives me the money, I have thousands of clones in the freezer where we know zinc finger talons, Cas9 cut at that position. What are the, it's for me, there is no drive behind that, because if you have an experiment that behaves like this, then I think so I have to step back. Human pluripotent stem cells, in my personal opinion, will have genetic drift. Human pluripotent stem cells, we don't have an assay for, does the cell still have the potential to be truly pluripotent, right? We can't do the experiment. So this is a tissue culture artifact. And so I am using it as a better HeLa cell. It's a much better HeLa cell. I would never touch a HeLa cell again, having this system now, okay? But it's still a sort of a HeLa cell. It's not a mouse HeLa cell where then you can test, does this still make an animal, right? So I'm going to do like this and look at telomeres and be a little bit naive about it. But if I had the money, I would be happy to provide, or even provide the clones for somebody to do it, because these are very well documented cell lines where we have hundreds in the freezer, genomic DNA. So if anybody hears this, they can have it anytime to just check that experiment. Yeah.
So it's a bit technical.
Yeah.
James Thompson group recently showed that they can reprogram a gene edits.
Yeah, it's a cool paper. I love that paper. So no, we haven't. So this paper, the paper that Jamie Thompson had a paper, you take fibroblasts, you Cas9 them, and then at the same time you reprogram them, and this Cas9 is editing so efficiently that the iPS cells that you are getting out of that are gene edited. There are two problems. He's also fighting with this problem of compound heterozygosis, that most of the iPS cells that he has have a designer mutation, but then are also broken or the other allele. Our system has a little bit of the advantage that, yeah, we are working with we have not used it. I love the paper, don't get me wrong. I think the key challenge in the field right now is to deal with making HR much more efficient, getting rid of NHEJ, which is solved, right? That's done, we know how to do that. Everybody can edit by NHEJ. HR is the challenge. And I think that is reflected in his results that many of the iPS clones have a hit on the other allele by NHEJ. Otherwise, the technique is, of course, fantastic.
SCR7, did you use it?
What's that?
SCR7.
Oh, is this one of these NHEJ inhibitors? I personally, we don't use it. So, of course, if you inhibit a pathway that is absolutely essential, right? You knock out Ku, cells are dead. DNA PK, there are hypomorphic cell lines reported, but they're very infrequent. So this is an essential pathway in humans. In mice, it's actually different, but that's okay. I am worried in a primary cell system to have this pathway suppressed. Because I also don't have a good assay to show that nothing happened to my cells. So this is maybe a little bit too conservative, and I should be more pragmatic. Once there is the golden bullet of how to make NHEJ go away for an hour while I'm editing, I will absolutely use it.
I totally appreciate the advantage of a big deletion.
Uh-huh.
So strategically, do you prefer to use a single cut?
No, do two cuts. So that is super efficient. Making a deletion where we have done this now. We have taken a promoter and cut it into pieces, and we started like 10 kb up, and then made 1 kb deletions, and then in some regions we made 100 base pair deletions. Completely easy. And there's a point about this. So about 60% of the junctions are predicted by the two PAMs between 3 and 4 being, so as you learned that the cut by Cas9 is a blunt cut, and it's upstream of the PAM between base 3 and 4. If you make a deletion, it's the 3 and 4 just come together in human iPS cells or ES cells with a frequency that we estimate based on hundreds, so be careful, probably hundreds of deletion events. 60% of those, the fusions is correct.